Surgeons do surgery. Plumbers fix plumbing. Neuroscientists study neurons. No surprises, it's right there in the name. Except for that last one. That's not always how it goes. In fact, this whole video is about a neuroscientist who doesn't study neurons. She studies the other brain cells, the ones that generations of neuroscientists completely ignored. They're called glia. And unlike neurons, nerve cells that send chemical and electrical signals, glia are cells that scientists once thought of as little more than the glue that held the neurons together. But Beth Stevens and her collaborators are showing the world that these cells may be the key to understanding Alzheimer's disease, and Huntington's, and schizophrenia, and glaucoma, and all sorts of stuff that we want treatments for. That's exactly the kind of story that inspired us to create this new series of videos called SciShow Field Trips. Stories that call for a trip outside of our comfy studio to see revolutionary science happening in the lab. So we sent our friend Jaida Elcock to Boston to meet Stevens and learn why she had no intention of studying the thing neuroscientists are supposed to study.
Thanks, Hank. Today we're at Boston Children's Hospital, which is just one of the many places Beth Stevens does her work. She leads a giant team of biochemists, computational biologists, psychologists, basically as broad a group as she can find. Or maybe I should say as broad a group, since the lab is affiliated with the Broad Institute of MIT and Harvard. The Stevens Lab thrives thanks to this diverse group of researchers, bringing their perspectives together to answer some huge questions about the brain.
There are so many reasons why having a group of people coming together that have different backgrounds is really what makes science really innovative and really exciting. I have immunologists in the lab, I have geneticists in my lab, I have computational biologists in my lab. It really enables us to come up with new ideas and to follow sort of new leads of science that I think are taking us into some exciting directions. Obviously, there are billions of neurons and trillions of synapses. It's very complex, right? But in the end of the day, if you were to zoom in on any one of those circuits in the brain, you'd see all these amazing connections and synapses. But underlying all those neurons, if you could then label all the other cells, including the glia, you realize that every one of those circuits contains all of those glia.
Right.
And they're also intertwined throughout the nervous system.
Stevens and her team think glia might explain why some of us stay mentally sharp into our 90s and others face severe cognitive decline, why some cells survive the brain's spring cleaning process and others are swept away, why everyone wants to park their car and have a yacht even though you literally can't drive in there. Okay, that last one's a joke. What I'm trying to convey is Stevens is a badass. But all superstar scientists had to start somewhere, and Stevens, like the cells she now studies, was a little overlooked at first.
I was just out of college. I had no idea what the NIH really was all about back then, but I showed up and I put in my resume and then nothing, because I had no experience really.
Right.
And so I had to then figure out how I was going to pay rent. So I got a job as a waitress, and I worked at Chili's, and I waited tables, and I carried fajitas over my head, and I kept putting my resume in every single week until eventually I got a cold call from who then became my mentor, Dr. Doug Fields. He was looking for a technician, and he somehow, found my application, and the rest was sort of history. I had an opportunity to work in a neuroscience lab.
Yeah.
And from there, I learned a ton, and he gave me a chance, and it kind of took me on this path to study neuroscience and then glial cells.
For more than a century after their discovery in the 1850s, glia had a reputation as the boring support crew for neurons. But Stevens and an early collaborator, Douglas Fields, found some pretty convincing reasons to care about them. Like the fact that neurons depend on glia to communicate with other neurons. You're able to think and move and taste and do pretty much everything you do because one of your neurons sends a signal to another neuron. If you can tell that you're looking at a screen right now, it's because the neurons that are sensitive to light can detect what's in front of you and communicate that information to neurons deeper in the brain that can make sense of the image. This communication from one neuron to another can take the form of electricity flowing along the long arms of the first neuron reaching out towards the second neuron. Those arms are called axons, and the electrical message traveling down the axon is facilitated by a layer of insulation called myelin. But myelin doesn't just exist out of nowhere. there. You have to make it, and that's where glia come in. They add the myelin to axons so that you can experience the world and function in it. But that also doesn't just happen. The complex dance of neurons needing myelin, asking glia to help them out, and glia adding myelin to their axons requires a lot of communication between neurons and glia. And that's what Stevens put her finger on in the Fields lab.
We sort of early on realized that, well, we shouldn't just study this from the neurocentric point of view. Like, what about, how the glia are regulating this process?
Yeah.
And I think that is really where I came into this. But initially starting to study the myelination process and how this is a really active communication going on between the neurons and the glia that enables that wrapping process.
Yeah.
But then later, also at the synapse itself, all these non-neuronal cells like astrocytes and microglia, they're also helping to build and also refine these synaptic connections. And the big question we have been asking is how? What are the signals? What are the mechanisms? And how does that work? Just like a wire is insulated, an axon, which can in some cases go the length of a spinal cord all the way up to the brain, so very long. Mm-hmm. So to make that signal go all the way in those long distances, it has insulation. And it turns out the insulation is made by the glia. They wrap their long processes around axons. And that is enabling these sort of electrical signals to move very efficiently. So those are myelinated axons, right? To some extent.
Incredible.
Right. So we started off talking a lot about that insulation. There's the insulation in a mouse brain.
Wow. I'm kind of thinking of it as like the neurons and the glia are both sort of electricians, and the neurons are like, hey, we need more electrical tape on this wire, and the glia are like, all right, I'm on it, I'll go get the electrical tape. Is that sort of?
That is one definite way of describing it,
Awesome.
And it makes a lot of sense. I think the other thing that we've realized is that insulation does more than just increase the efficiency of a signal. Those glial cells release a lot of important signals that keep neurons and axons healthy.
Okay.
And so when that doesn't happen anymore, that can also lead to, these brain or neurodegenerative conditions that can lead to unhealthy synapses and axons and neurons.
Yeah. Her discovery of glia's role in the myelination process kick-started her career. It turned Beth Stevens into Dr. Beth Stevens. But that was just the beginning of her fascination with these overlooked cells. As she told colleagues about her research, the NIH director of the National Institute of Neurological Diseases and Stroke personally encouraged her to keep heading down that road. This led her to the lab of a trailblazer in glial research, Ben Barres.
When I was still at the NIH, I was studying this process of myelination and realized, wow, it's not just sort of the support of glia. There's this dynamic interactions and crosstalk going on along those long axons. At that time, the Barres lab was studying this dynamic crosstalk at those synaptic junctions.
Okay.
So that's where I'm like, wow, I want to learn more about that. I want to learn more about how synapses are forming, how they're developing, how they're being remodeled.
Ben Barres was the guy who discovered that glia are pretty badass in their own right. He showed everyone that glia can communicate using neurotransmitters and other molecules. So they were much more interesting than almost everyone thought.
Neurons release signals to the glia that say, okay, it's time to do something. And similarly, the glia release signals that tell the neuron it's time to do something.
Okay.
So we should be thinking about this as a two-way dynamic conversation between the non-neuronal cells and the neurons.
By the time Stevens joined the lab, neuroscientists knew the basics of how the brain sets up neuron-to-neuron communications. A bunch of neurons make a bunch of axons. Some of them get myelinated by glia and become stronger connections, while others are removed so you don't waste your resources on them. This removal of excess inputs is called pruning, and it works just like pruning a tree.
The idea is that, you sort of have sort of an excess of connections. And that's actually probably good because you sort of have extra connections there ready to go. But then some of those connections become meaningful, right? Where that's going to be important for a particular action or particular cognitive function. Some of those connections get strengthened, the ones that are meaningful, and the less relevant inputs get pruned away. So it's this sort of like, use it or lose it idea.
At that point in time, neuroscientists knew that you needed pruning, but they didn't fully understand how it worked. Stevens realized that there might be a hint in the world of immunology, where cells called macrophages act as a kind of cleanup crew in the body.
There's a lot known about how macrophages work. They help defend our body against things like infections, right? They are really good at engulfing or removing unwanted cells, debris, including a bacteria, for example. So they do this by recognizing molecules like they call eat me signals that essentially coat the surface of that cell. And through receptors on their surface, that enables them both to recognize and actually engulf. And almost every tissue in your body has macrophages.
Interestingly, some of those eat me signals were showing up in the developing brain, but only on some neurons. The signals were complement proteins, which is a term borrowed from immunology.
So it turns out that there were a lot of these complement molecules in the healthy brain, and it also made us realize, well, the brain has a macrophage called microglia that had very similar receptors that recognize these complement molecules. So that led us to wonder, instead of removing a pathogen or unwanted debris in the body, could they be playing a similar role in removing these extra synapses during development?
Right.
And that's really what launched a lot of the work I did as a postdoc in the Barris lab. And it really began the foundational work that launched my lab here. This is an example of one microglia in green, and now you're looking at some synapses. So every one of these bumps, or so the ending of an axon or a synaptic input, some of them are touching, and we can also stain the brain sections, not only with synapses, but with these eat me signals like complement, which is what's shown here. So in this case, it looks like the red is a complement molecule called C3, and the green is a synaptic marker. And you can see, if you were to zoom in, you can see that there's almost like a tagging of subsets of those synapses.
The microglia are kind of our gardeners of our neurological garden to make sure that we have everything looking pretty and working properly.
Exactly. What we're finding is that the microglia help to do this sort of fine-tuning and pruning and remodeling of connections. One of the ways that pruning happens is through literally nibbling off and eating, which is what we've been studying with complement.
So as it turns out, microglia play a really important role in pruning during healthy development. But by the time you're an adult, that process is supposed to shut down. When Stevens published some of her findings in 2007, she hinted that pruning might be a problem later in life.
Could some of these same mechanisms we've been studying in the healthy developing brain become kind of almost like reactivated? If you look at a couple of animal models of Alzheimer's, do we see complement tagging those vulnerable synapses? And indeed, very early on, using multiple different models, we saw some evidence of these sort of eat me signals. And those microglia that have all these other roles beyond pruning also started looking a little different, and they had evidence that they might also be kind of engulfing these synapses. So we said, if we block the eat me signals, or the receptors on microglia that recognize, that enables them to engulf and nibble off these synapses, is that a good thing? Can we protect synapses? And if so, does that, at least for a mouse, does that help them, to do better cognitively? And the answer, at least in these animal models, was yes.
Suddenly, glia were at the center of the pruning story and connected to one of the most challenging diseases of the brain to study. So how did other neuroscientists kind of respond to these findings about glia? Were there a lot of people talking about these at neuroscience conferences?
Back when I started out, no. I would say if I think about a typical neuroscience conference, there were very few talks or posters that kind of focused on, neuroimmunology back then. So when we started initially sort of publishing our work, there were definitely questions and almost some skepticism that this was going on.
So if you're a neuroscientist with an underappreciated appreciation for glia, where do you go? You give a speech at an immunology seminar.
I can remember being invited as a new faculty member in a neuroscience department here at Children's Hospital to give a talk in the big immunology lecture series. I think I'd only been in the lab about a year. I was petrified, completely petrified, because I had never really formally studied immunology. Of course, I've read a lot, and I learned a lot about complement, for example. But the idea of getting up in front of this group of experienced immunologists and then presenting this work was very sort of unnerving, if you will. But I decided to do it anyway, and it was probably one of the most important lectures of my career, not because of the talk itself, but because I communicated the same story I'd been giving to neuroscientists, But the questions I got and the ideas that stemmed from those questions were really unique to the immunology community. They were seeing connections that I could not have possibly have seen based on their own experience and their own expertise in immunology. And that sparked lots of new projects in the lab, lots of new questions, new collaborations that continue to drive our research.
That talk essentially launched the Stevens Lab, which started with two main projects that she wanted to investigate. One was to continue basic research into how microglia and those complement proteins operate in the brain. The other was a project applying all of these new findings to Alzheimer's disease.
So, as Beth mentioned, we're very interested in what we call the borders of the brain. And the particular border that we're studying here is the outermost layer of the meninges, or the dura mater. And the really important thing about the dura mater is that it's a key immune hub for the central nervous system. So if we take a look at the screen, we have a skull cap from a mouse. Our goal here is to actually extract this meninges in one piece so that we can better study the types of immune cells that are there. And I'm just gonna start by kind of grabbing onto the skull cap here and starting to cut around using this small pair of scissors to just trim the skull down to kind of a consistent size like this. So now that I've cut the skull, I want to go back and carefully start to peel the meninges out of the skull. But we can start by kind of grabbing the meninges at the edge of the skull and starting to peel it away here. Again, being very careful not to scrape the bone too much. And so, once we were able to extract the meninges, we study it in many different ways. We do a lot of imaging on the meninges. So we actually can take this whole piece out, stain it again with different antibodies, and learn more about where these different types of immune cells are and with whom they may interact.
Stevens also makes sure that the people working on the two big projects in her lab are talking to each other regularly, because she has seen how much of a difference that can make for breakthroughs.
The work I talked about on complement was one example where I clearly could benefit from an immunologist to kind of help us think more about how to study these immune molecules more deeply, but also provide new perspectives and new ideas, new techniques and approaches that immunologists use that neuroscientists might not think to use.
By learning about glia and Alzheimer's in tandem with each other, the Stevens Lab is making huge discoveries today.
There are already some good biomarkers in Alzheimer's disease. A lot of focus has been on this amyloid, this toxic protein in the brain, and tau, another misfolded protein that can be very harmful to neurons and synapses. But what it doesn't capture is what's happening to the immune cells and the microglia. Could they also be combined with those other markers to try to then bring the microglia into the picture? To be able to both stratify patients, try to understand where they are in disease progression, and many of these patients have multiple different pathologies. There's not, at this time, a way to be able to disentangle that. So a lot of the work going on in the lab, but also in the field, is to try to translate some of the findings that we're uncovering here into new biomarkers that one could read out in a non-invasive way to see how these microglia are changing across disease in humans.
But the reason glia research is so groundbreaking right now isn't just for their involvement in Alzheimer's. They're such fundamental cells in your brain that understanding them better has huge potential to explain all sorts of diseases.
So it's not just Alzheimer's disease. There's now evidence to suggest that some of these immune and glial-related mechanisms could be relevant across many other brain disorders as well, ranging from ALS to Huntington's disease, Parkinson's, and others. So there's a lot of people in the field become increasingly interested in the immune system and in glial cells. In this case, because the genetics are really pointing right to them.
And more we're finding that there's a desire and an excitement to start working together, because what one person found in one experiment in their lab may very much relate to what we're interested in, but if they don't know what we're talking about or working on, how would they ever connect the dots?
Exactly.
So we're really starting to realize if we're going to tackle really hard problems like understanding neurodegenerative disease or brain development, we need to team up and work with folks in many different contexts, and I think that's going to change not only how rapidly we make discoveries, but also much more fun to work with people that are sharing their insights and data.
Yeah, absolutely. So are the tides changing a little bit? Are neuroscientists, becoming more interested in glia?
Oh, yes. I think that is just so much more. If you walk into, let's say, one of the biggest neuroscience conferences in our field, is international. It's called the Society for Neuroscience meeting. And it used to be, when I was starting out, you could search for glia and you would see a few talks, a few rows of posters. Fast forward now, 20 years later, you search Glia, it's in every row, every talk, one way or another, even if it's not the main title, it's coming up.
Right.
Because, you know, glia are part of the brain, not surprisingly. And neuroscientists and glial biologists are no longer separate fields. I think that is more and more evident now than ever before, and I'm so happy that's the case. There's just no end to where to take this because there's so many unanswered questions. A lot of them relate to glia.
Thanks to Stevens and other like-minded researchers, the cells nobody cared about are now widely recognized as the key to understanding some of the brain's biggest mysteries.
SciShow Field Trips are made with our friends at HHMI Tangled Bank Studios. We've come together to bring you face-to-face with researchers at the cutting edge of scientific discovery. You can watch more of Tangled Bank's science content at tangledbankstudios.org.